AMALearning, you're about to. Specification Point 1.16 Core Practical. Investigate osmosis in potatoes. To successfully answer any GCSE question linked to this specification point, you will need to know how the concentration of a potato cell can be determined. How can we determine the concentration of a piece of potato? Step 1. Cut tubes of potato using a cork borer and trim to 5 centimeters. This ensures that the size of the pieces are controlled. Step 2. Record its start mass. Step 3. Place the cylinder in a boiling tube of 0 molar solution and leave for 20 minutes. This controls the time and allows enough time for osmosis to occur so mass change can be measured. Step 4. Remove the cylinder, blot dry and reweigh. Blot dry means to remove the surface water. This prevents any excess water being left on the cylinder. Step 5. Repeat for different concentrations. This gives us a range of data that we can use to determine the potato's concentration. Step 6. Repeat twice more. This means we can identify outliers which can be discarded before calculating an average. Step 7. Calculate the percentage change in mass. All the potato pieces would have different starting masses, and this enables us to do a fair comparison. As 1.16 is a core practical, you may be asked about the potential sources of error in this experiment. The following are potential sources of error. Inaccuracy in the balance. Cylinders dried a different amount. This is a common mistake. Students overblot or dry the cylinders. This leads to some of the water that has osmosed into the potato cylinder being removed. Evaporation from tubes. It is advisable to cover the test tubes with some plastic wrap or a bung. Inaccuracy in the concentrations of the solutions. Clearly labeling the test tubes helps to avoid this error. How can we use the results from this experiment to determine the concentration of a potato piece? Step 1. Calculate the percent change in mass. Step 2. Plot the percent change in mass against concentration. The independent variable, the concentration, goes on the x-axis, and the dependent variable, percent change in mass, goes on the y-axis. Step 3. Draw a line of best fit. The point at which the line crosses the x-axis is the concentration of the potato cells. This is very important, so let me repeat this. The point at which the line crosses the x-axis is the concentration of the potato cells. Why does the potato gain in mass, or why does a potato lose mass? This is a very common exam question. Let's initially look at why the potato gains in mass. You must mention the following to get full marks. Water enters the potato cells due to osmosis. This means water goes from an area of high concentration, this is referring to the water outside the cell, to an area of low concentration, the water that is inside the cell, through a partially permeable membrane. Let's now look at why the potato loses mass. You must mention the following. Water leaves the potato cells due to osmosis. This means water goes from an area of high concentration, in this case this means inside the cell, to an area of low concentration, which is now referring to outside the cell, through a partially permeable membrane. Our animations were designed by experienced UK-trained teachers. We have been achieving amongst the best results in the country for many years, and we decided it was time to share our success. Have you just amaled and want to amal some more? Don't forget to like, subscribe and share to keep yourself up to date with the latest uploads. AMALearning, you just did.